Okay, folks, let's get started. Hi, everyone. Welcome to Tuesday Night Rheumatology. This week, like last week, we're doing a Still's Disease Journal Club, and I'm joined by some real experts in the field. If you all would turn on your cameras and whatnot, that would be fabulous, and we can introduce ourselves to the audience. I'm joined by Olga Petryna, Hermine Brunner, and Dan Lovell. Dan's camera's not working well, but I'll ask everyone to introduce themselves and where you're from before we get started. Start with Olga.
Hi, I'm Olga Petryna. I'm a rheumatologist at the Scarsdale Medical Group in New York area.
Okay, and Dan?
Dan Lovell. I'm a pediatric rheumatologist at Cincinnati Children's Hospital.
And Hermine, turn on your microphone.
Hermine Brunner. I'm a pediatrician, a pediatric rheumatologist at Cincinnati Children's. And like Dan Lovell, I was one of the lead authors and lead investigators on the systemic JIA studies that are being reviewed today.
Superb. Okay, folks. So what we're going to do here is I'm going to do a share screen, and we're going to review for our audience what we think are two important papers regarding the treatment of pediatric and adult Still's disease. And we've chosen these because all of the discussants have a fairly good history with the data and these studies, and we'd like to get their perspective. I want to ask the audience that if you have a question, don't use the chat, use the Q&A, and we'll get to that during or after the Journal Club. We will end at the top of the hour. In the chat box, however, you will find links to the articles that we're presenting today. So let's start with this opening slide that I have here. As you can see, this is sort of the introduction to what we're doing and who's on the call. We're going to talk about a canakinumab trial used for the approval of in adult Still's disease, and the TENDER trial about tocilizumab and its approval that led to its approval in systemic JIA. So let's begin by, I want to just make a few comments here that I think can help set the stage about where rheumatologists are. Now, this is biased in that it's largely a survey of adult rheumatologists. In 2018, 250 rooms were asked about what drugs they use and what they think. 70% believe that Still's is an IL-1 driven process, and a quarter believe it's an IL-6 driven process. When they were asked about after steroids, what were they likely to use, half would use a biologic and half would use methotrexate. When asked which biologic, 71% preferred anakinra, 15% preferred tocilizumab. And you can see that 20% of the people responding had no Still's patients and hence no experience. And the rest did have some degree of experience, but most of them were getting their patients referred from either PCPs and hospitalists, infectious disease, and very few were actually de novo new cases to them. But I thought this was important. So we're going to review two main treatments here, and we're going to be talking about both systemic and articular disease. But you can see that the leading candidates here are going to be anakinra, canakinumab, and tocilizumab. And the ACR guidelines on the treatment of systemic JIA recently published a few months back. And they actually reviewed both treatments of oligoarthritis and TMJ, but then systemic disease. And here's the algorithm. And they first asked the question, if you have MAS, you go somewhere else. But if it's systemic JIA and you don't have MAS, then if you do have MAS, they're recommending you could treat with an IL-1 or IL-6 inhibitor as first-line therapy. And the light green boxes are conditional recommendations. The dark green boxes are more evidence-based. And then they also say and/or trial NSAIDs, because there's this historic thing about NSAIDs and their use in systemic JIA. But I think, the question is now, do we go right to IL-1 or IL-6 inhibition? Currently, drugs that are approved for use in systemic JIA in kids, canakinumab was approved in 2013 in the United States. Earlier, tocilizumab was approved in 2011 for the IV administration, and then in 2018 for the sub-Q administration. And then only in the last 2 years has canakinumab been approved for use in adults with Still's disease, and that's sort of a new development. But, you know, everyone tends to talk about anakinra. I'm going to show you 2 slides on anakinra because most of us that do use cytokine-directed therapy still are using this as opposed to the 2 drugs I just showed you. Here's 2 studies, one from Peter Nigrovic from Boston, 46 patients, a multi-center study, 60% having a complete response, 39% having a partial response, generally good data. And this is like what a lot of the trials look like early on. And these were trials actually that both Hermine and Dan were involved in. Another study just more recent from Fabrizio Di Benedetti showing of his 56 patients that were treated in trial, looking at remission off steroids, that 73% met the criteria for clinically improved disease at roughly at 3 months is when this was achieved in most patients. So anakinra, there's a lot of both open label, but also some control data saying it works, especially in kids, but very little in adults, I must say. So, and then more recently in 2020, the FDA came out and said that canakinumab was approved for treatment of adult Still's disease based on the data I'm going to show you, noting that it had already been approved for systemic JIA in kids over the age of 2. I don't know, prior to this approval, I was getting this drug approved by saying my patient, who's 37 years old, is over the age of 2 and therefore merits treatment with canakinumab. And yes, he does have systemic JIA. It just happens that he's an adult. And, you know, Nikolov, who is the division director at the NIH, said this is basically a brand new and important approval for patients who have no options. But more importantly, the FDA release has this line, which I think is important and bears mention here, in that AOSD is a rare and serious condition of unknown origin. And basically, he equates adult Still's disease with systemic JIA and suggests that this is a disease continuum rather than these being 2 distinctly different disease entities. So the FDA coming out and making that, taking that stance, it took them a number of years to do that, but it was nonetheless important in getting canakinumab approved in adults with Still's disease. So I'll stop there and ask for any comments from the discussants on the call.
Well, I think I would agree with defining adult Still's disease as a continuum of SJIA, because we do see a lot of common clinical features. We see similar interleukin signature in both conditions, and the patients tend to respond to treatment similarly, to similar groups of medications, I would say. So there is like a lot of similarities in presentation. And of course, as a person grows up and progresses in their life, some of the manifestations can be different. That's why maybe other physicians could think of it as a different condition when we diagnose it in adults. The other thing I would point out in my experience is a lot of patients who are diagnosed as adults, they actually had symptoms in childhood, but they were omitted or not diagnosed as a childhood disease. So the question is, are all adult Still's patients truly adult onset Still's disease, or are they the delayed manifestations of childhood disease or relapse after prolonged remission?
Right. One of the key features that we've learned from our pediatric rheumatologists is that this is a disease that has disease-free intervals that could be long, could be months, could be years. And as Olga points out, the series of adult Still's shows that somewhere between 25 and 50% of patients had a prior illness in childhood that could have qualified as systemic JIA. Hermine, what's your feeling on that idea that this is the same disease in adults and kids?
I think it's the same disease, both in terms of involvement and response to therapy. Albeit in children, there must be some larger genetic contribution to the disease that then elicits the early onset of disease during childhood.
Again, it'd be great to know what those triggers are. Is it a genetic predisposition? Is it an environmental trigger? We don't really know. What was curious is that some of the studies that have been done on mainly systemic JIAs show that many of them have some of the genes that predispose to other periodic fevers. And that sort of links to maybe there's a genetic predisposition. You know, could also mean that maybe they were misdiagnosed. But Dan, what do you think?
No, I agree with the others that it's the same disease. Number of studies have shown that that's the case. Another learning that we're doing in pediatric rheumatology is we have to take systemic JIA out of the mindset of JIA because it's really tremendously different than the other. As you pointed out, it's an autoinflammatory disease, and all the other types of JIA are autoimmune. So our current classification criteria is undergoing review to more appropriately put systemic JIA with other autoinflammatory diseases and take it out of the subset of autoimmune diseases. So that's another change we're going through. And I think there's an international group convening to come up with similar diagnosis and response criteria for the full spectrum of disease in adults and children.
Right, and that would be great to see because it's sorely needed, as these trials will tend to point out. So the first is the canakinumab. I'll try to go through these somewhat quick because I'd rather hear your comments on these data and their applicability. This study was published in 2020 and kind of was the basis upon which it was approved in adults. And that's surprising when you see some of this data. Again, you should note that canakinumab was EMA approved in Europe back in 2016, as was anakinra. But we had no approval for adult stills, and then until this data came along. In this trial, there were 56 patients that were enrolled, all of whom were adults, all of whom had active disease, at least by arthritis, with greater or 4 or more tender and swollen joints. They all met Yamaguchi criteria for the diagnosis, which means they must have had more than just the arthritis. And then they were randomized to receive either placebo or canakinumab with a max dose of 300 milligrams per day every 4 weeks. The endpoint here, which is one of the problems in Still's disease studies, is been clinically relevant reduction in disease activity as measured by a change in -28 of greater than 1.2, meaning we don't, as Dr. Lovell just pointed out, we don't have an endpoint measure that's specific for systemic disease. And as was done in the Tender trial, it was improvement in the JCR30 with no fever. Now, they also looked at no fever in these patients, and we'll show you that data. You could see on the left the sort of confused nature of the enrollment here. 41 patients screened, 36 randomized, 2 were excluded, 1 with Whipple's, and there were also 2, I guess it was one of the ones with Whipple's, that that were excluded because they were supposed to be in the placebo population, and then at week 4 they got the canakinumab infusion, and in the end we're looking at, you know, the 17 on canakinumab and 14 on placebo and how many responded. And you can see of the 17 canakinumab, there was 12 out of 17 responded. In the placebo, of the 14, it was half of them responded. And then on the left, you can, your right, you can see what these patients look like, meaning there were very few, 33 to 47% with disease of less than 1 year. 70+% had received a biologic DMARD. 66 to 76% had previously received anakinra. A third had received TNF inhibitors prior to the trial, and 10 to 20% had received tocilizumab. So you can see this is a fairly experienced population as far as drug exposure, and meaning that they were really quite refractory. Yet, you know, 9 and 8 had fever going in. A few had lymphadenopathy. 10 total had skin involvement. Myalgias was in 1 patient. So they did collect that. They did look at that. Turns out that the systemic features were not very important overall here. Olga, any comments about the enrollment criteria?
Yes, I think what it brings us to is what type of Still's patients we are actually treating, or which type of patients with Still's were chosen for this trial. It seems like the duration of the disease and manifestations point towards most chronic articular Still's rather than systemic Still's that we typically see in the beginning of the disease course. And that makes me think about what type of interleukin inhibitor would you choose for an early patient with more systemic manifestations as opposed to patient with chronic articular disease. And it may even signify that the interleukin signature changes over time, depends on what manifestations patient has. So I think it also influenced the results of the study, even though it wasn't complete, it was terminated early, mainly because the category of the patients were predominantly articular.
Right, right. And it could have been articular because a lot of them received systemic therapy that could have washed away the systemic features and left them with chronic articular disease.
Correct.
And while we could say it's disappointing that they weren't treating systemic disease, we could say, well, interestingly, the chronic articular patients that didn't respond to all those other therapies did respond to canakinumab, but let's see how much. So I guess it was, the numbers are a little wrong here. It's 37 were picked to randomize. One was excluded, so it's 36, but then 2 were excluded later on. And that's the basis for the analysis. They had high disease activity as far as their DAS28 ESR scores being over 5.2. And the intention to treat analysis, they had 67% in the canakinumab group versus 41% in the placebo group who responded in meeting the primary endpoint. And that was, again, a change in DAS of greater than 1.2. That was not significant, meaning the primary endpoint was not met. All right. And that difference between those two groups was not significant. While they measured systemic manifestations, there was no significant changes in those, but that's probably because there were too few. At week 12, the number of canakinumab patients that was free from fever for at least 7 days was 78% versus 65%. Again, a trend, but not significant. Same thing was less skin manifestations with canakinumab versus placebo, 4.8 versus 6.7. Again, a trend, but not significant. After they excluded their 2 patients, did a per protocol analysis. They said that there were greater ACR 20, 50, 70 responses. I'll show you that in a second. And the number of people who had DAS remission was 33% with canakinumab and 11% with placebo. Again, not statistically significant, but the numbers are low. The group size are low. On this slide on the top, you can see what happened to ACR 20, ACR 50. Just look at the responder columns to see the numbers. The ACR 20 was 61 versus 41. The ACR 50 was 50 versus 17. The ACR 70 was 28 versus 12. But none of those were significant, but I think our sample size here is really quite small. They had patients who entered open label. They all did well, but they only had open label accounting for the 7 responders who went into open label. And then their safety outcomes were really quite mild, and with a few cases of SAEs that were seen, none of which were clearly related to the drug, and, there's a lot of intercurrent illness kind of things going on here. So again, they came away saying, apologies. First off, the article says that, they made the bridge between systemic JIA and adult JIA, systemic JIA. And then they said they had problems with recruiting because of the rarity of disease, the severity of disease, and the EMA approval of canakinumab. It made it hard to recruit. And that's why they prematurely terminated the trial with only 36 out of their plan 68 patients needed for their efficacy analysis. That was further hampered by not having identical placebo, and that led to some basically procedural problems that led to some mistakes in dosing. They did show in a sub-study that looked at biomarkers that the transcriptomic signatures in adults was similar to that of systemic JIA, and I think it's based on this positive data, along with previously positive data in systemic JIA, that it gets the approval in adults and becomes the first biologic approved for adults with Still's disease. But I want to ask Hermine and Dan what their opinion is. Obviously, this data is not strong, but is it strong enough given what you know and what you and your colleagues have proved with canakinumab and other IL-1 inhibitors in systemic JIA?
Well, over here, the premise is that adult onset, or the stance of the FDA was that adult onset Still's disease is the same disease as systemic JIA. Different than what we normally see in rheumatology trials. In SJIA, 3 trials were done to get approval for canakinumab in systemic JIA. And this time, the adult onset disease followed up and canakinumab was approved in the adult onset Still disease population because of the good experience in systemic JIA and by virtue of extrapolation of the evidence from children. So we saw similar trends, we saw similar improvements in adults as in children. And as you pointed out, the results were not significant, but that, the study was largely underpowered.
Right. Okay. So with this data and with this approval, I guess I want to ask Olga. Olga, has getting an approval for your adult patients made life easier in treating your adult patients? Are you more able to go to biologic therapy by having an FDA-approved drug?
Absolutely. I think it makes it much easier to approve than it used to be in the past. In the past, we had to be creative, call it as periodic fever syndrome, you name it, just to get insurance to approve the medication, even show value of other treatment options. But now with this approval, and this being pretty much the only FDA-approved medication for the condition, we can argue with insurance companies when they insist on, let's say, methotrexate or cyclosporin or whatever, another crazy request they make, that those are experimental treatments for the condition, while canakinumab is FDA-approved, and it usually makes the process very easy. So even if it does not get approved from the first request, on the appeal with the letter of medical necessity, it makes it very easy to get the medication to the patient.
I want to quickly go back to this slide and ask Dan and Hermine their opinion of this algorithm by the ACR that says that if you have systemic JIA and there is macrophage activation syndrome, MAS, and you go to the right, the first recommended therapy is IL-1 or IL-6 inhibition and or glucocorticoids. Is that the standard of care? I mean, your center in Cincinnati has been a leading center in not only in systemic JIA, but also the MAS and our understanding of that. Is that where we should all be going when we encounter macrophage activation syndrome? Dan? Hermine? Oh.
Well, we know that macrophage activation syndrome very likely is associated with hypomorphic mutations that are also seen in HLH. So in hereditary histolymphocytosis. And we do know that one of the triggers of MAS is high disease activity. So treating systemic JIA effectively with IL-1 and IL-6 inhibition helps controlling the MAS. Now, for another treatment for MAS is systemic steroids. They're not always sufficient, but they are the first-line treatment for MAS. And so if there's a high inflammatory state, MAS, systemic steroids would be indicated. Having said that, it has been shown in the past that neither IL-1 nor IL-6 inhibition can completely abort the development of MAS episode in those who are predestined or who are genetically predisposed getting it. There's increasing evidence that the MAS process is driven by interferon gamma and high levels of them. And for those, very often, JAK inhibitors are used as co-therapy.
Interesting. Have you, I know you know the data of Fabrizio Di Benedetti about emapalumab, the monoclonal antibody against gamma interferon. Do you have experience with that?
Yes, it works like a charm. So this MAS is really a spectrum. Some children get maybe one episode, once at the beginning of the disease. Some get them regularly. Some have chronic MAS. So the MAS never withdraws and can actually cause or is associated with interstitial lung disease and other organ damage, including chronic liver disease. And there are some who are so severely involved that, they are basically close to dying. And we had a couple of kids who had very high inflammatory markers with ferritin levels in the 30 and 40,000. And with emapalumab, those levels came down, and these children actually now have a normal life, but they do need intermittent or sometimes even chronically emapalumab.
Nice. Oh, so I like
And the
that.
response was really quite dramatic. We had kids sent to us, went straight to the ICU, and within a few days, a couple of doses there, they were tremendously improved and went home in a month with minimal steroids where they had been steroid dependent and still doing poorly. So the response can be quite dramatic. Jack, I think my only addition to this slide would be to think more severe cases of MAS in which you don't necessarily have access to that biologic. Cyclosporin has been shown to be very effective in treating MAS too. So the combination of systemic glucocorticoids and cyclosporin are very effective treatments for the vast majority of people with MAS, systemic Jiaa patients with MAS.
And when you consider all those things, the steroids, the cyclosporin or etoposide, or the IL-1 or IL-6 inhibitor, or emapalumab, speed becomes more important the sicker the patient, because these kids, these adults can have up to a 40% mortality rate.
Right, and it's about 20 to 30 in children, so.
Yeah, okay. All right, excellent. All right, so we're gonna go on and look at the data from the TENDER trial. Here we go. So the TENDER trial was a pivotal study. I think it was 2012 that this came out. You know, a lot of these trials are done by Printo and PRSG. I want to ask who wants to tell me what this is. Obviously a collaborative study group that's highly productive that you're a part of, but can someone address the group and what they do?
So the PRCSG was founded probably over 45 years ago, well before the proliferation of eponyms and that sort of thing for network names. So it's Pediatric Rheumatology Collaborative Study Group, and there's centers throughout North America for the PRCSG. And then Printo is a more recent group, but covers the rest of the world. And so they started in Europe, and now they spread to South America, Asia. They've recently added centers in China and India. So it's truly between the two groups, they cover the globe, and we have shared responsibility for designing and performing these studies now collaboratively for 20 plus years. Yeah, and the goal is to do a single study that stands for registration for kids with that form of JIA throughout the world.
Yeah, it's highly commendable. And anyway, so this study, which I thought was a game changer as soon as I saw it at ACR and EULAR those years, 112 kids age 17, I'm sorry, let me go back here, sorry, age 2 to 17 years had to have active systemic JIA for more than 6 months, despite treatment and an inadequate response to nonsteroidals and steroids. And then they're randomized to receive the anti-IL-6 receptor antibody tocilizumab at either 8 or 12 milligrams per kilogram. Again, the adult rheumatologists are well used to the doses of 4 to 8, not 12, but 12 is required, especially in kids under 30 kilograms, or they were given placebo. And these infusions occurred every 2 weeks for a 12-week period. And that was the primary endpoint. Of course, the study went on for 2 years after that. Overall, there was 37 who were randomized to placebo and 75 to tocilizumab. The primary endpoint was a JCR 30, that's like ours, 6 variables with a certain number, 4 out of 6 needing to be over 30% response. And there are articular variables, and then also not having fever. That's an important add-on here. And after the primary endpoint, non-responders were certainly all open or offered to receive tocilizumab as open label therapy. This is the profile of patients enrolling in the study. The average age was 9 to 10. The disease duration was 5 years. You can see that 70% or more received a DMARD, most of that being methotrexate, but even cyclosporin, thalidomide, and sulfasalazine. 78 to 84% had previously received a biologic agent. 35 to 55% had previously received an IL-1 inhibitor, and 70+% had received a TNF inhibitor. Another, I believe, a gigantically common mistake in rheumatology to treat systemic JIA or adult stills with a TNF inhibitor if they have systemic features. Many of them were on methotrexate during the trial, and many of them were also were on glucocorticoids, as you can see here. 80% or so were stayed on their glucocorticoid dose. This was one of the earliest slides that I made. This is back in 2010, that showed the number of patients responding. The primary endpoint of a JCR 30 was 85% versus 24%. And then in the open label extension, they did continue to do really well. On the bottom, you can see how many were fever-free, how many were rash-free, how many had met the JCR 70 definition. Again, highly impressive data that was seen in this trial at 12 weeks. Here you can see what happens to their the JCR 30 and the other ACR 70, 50, 90 responses on the right. Obviously, those that are on drug are the top three lines. I'm sorry. This is actually the response rates beyond week 12, and the lowest line is at ACR 90 with no fever. So all these joint responses are predicated on not having any fever going out to 2 years. And then you can see similarly that as they get better, their obligation to use glucocorticoids goes down significantly, especially after week 12, and sort of really being a very low number by the week 52. Any comments on these response rates?
What?
Okay.
Yeah, I'm trying to get unmuted here. So Jack, can you hear me?
I can hear you. Yeah, go ahead. Dan.
So I think if you look at the response curve, that graph on the left, you can see how rapidly these patients respond. So, this is given once a month. So you can see by after the first injection, over 60% of these patients demonstrate an ACR 30 and no fever by the week 4 visit. So the response in these patients is really quite rapid and dramatic. The higher levels of response, a more articular improvement, take a little bit longer, but the ACR 30 and no fever is really very rapid. And the ACR criteria, only 2 of them articular. One is SED rate, and the other are the parent global of overall disease impact in the MD global of disease activity. So it's a very comprehensive assessment of the state of the child with the ACR 30, 50, 70 parameters. And then you add the control of the systemic features. So these kids were, as you showed in the background, chronic non-responders who had been tried on everything available. They were tried on TNF because, at this time we had no approved IL-1 or IL-6 therapies. And so these kids had demonstrated persistent disease. Over 50% of them still had active systemic features at the time they were enrolled in the study, on average 5 years later. So this was a very treatment-resistant group, and you can see what happens when you treat them with a biologic that tightly fits the biology of the disease.
Yeah, it's amazing. Do you think the rapid response was the result of the Q2 week administration of tocilizumab, or was it the doses that were used, which were admitted, I think, really quite high, including a subset that needed the 12 milligrams per kilogram dose?
Well, so they did PK assessment. I misspoke. It's every 2-week dosing. I'm sorry, but they did PK assessment after the fact, and the exposure for the younger kids, the smaller kids that are on the higher dose, is the same as the other group. And we did some earlier PK work in this group and adjusted the dose based on what we felt was necessary to control the systemic features, because systemic disease, at least systemic JIA patients, the levels of the inflammatory cytokines are really much higher than they are for polyarticular disease. So the dose of tocilizumab for systemic is higher than it is for polyarticular JIA. And we did that to mirror the fact that the intensity of the inflammatory response is greater in kids with systemic JIA.
Yeah. Again, these are very impressive data. We only wish to see this in our kids, but this table shows you a comparison of responses, and you can see just the first line, the JCR30, which is 24% in placebo and 85% in tocilizumab. But I want you to go down and look at the systemic features, that those also improved. You have a baseline value where the fever and the rash, they didn't get better on placebo. In fact, they got worse. But on tocilizumab, the fever went down from 55 to 15, and the rash went from 29 to 36. And then the acute phase reactants and the labs also showed dramatic improvements, suggesting, again, the efficacy of IL-6 inhibition in systemic JIA. The adverse events here, I didn't find a lot to talk about here, although I do want to talk about this one line that's midway down here, where it's macrophage activation syndrome, about 2/3 of the way down on the left. You can see that no cases were observed in the double-blind portion of the study, but in the extension, 3 cases of MAS occurred while on tocilizumab. And I'm sorry about that. And my question to the group is, and Dr. Brunner said it earlier, that not everybody will do great on IL-1 or IL-6 alone if you have MAS, and this data says that you could get MAS on an IL-1 inhibitor or an IL-6 inhibitor. Anybody want to comment on this particular adverse event, or any of the adverse events for that matter?
We actually looked at MAS events, and the frequency seemed to be not increased by IL-6 or IL-1. And these MAS events were very often triggered by an EBV infection. And the MAS episodes were easily treatable. I would like to point out about the prior slide you showed, that these response rates in SJIA on tocilizumab were seen even under consideration that steroids were tapered starting week 8 of the study. So what you see here as lines and improvement over time, would even be more rapid if steroids had been maintained at the doses at baseline. So MAS occurred, and and it's, as I stated, there's a genetic predisposition, and as you cannot change the genome of a patient, you will see MAS episodes in those patients.
Hermine, can I ask you, if a patient has MAS in the face of active systemic JIA, are they likely to get it again in the future?
Very often.
Okay.
That's the strongest. That's the strongest predictive factor for recurrence, is the first case. So yeah, I'd say it's about 50% get recurrence.
Yeah.
And I think both in this trial of IL-6 and also in the trial of IL-1 in systemic JIA patients, we did see MAS. And so great disease control is not a get home free card for MAS. You still need to have a high index of suspicion. And as Hermine said, we've looked at it closely, and treatment with these drugs does not seem to increase your risk of MAS. But it's certainly, you need to continue to have a high index of suspicion for MAS, even in children whose disease is well controlled on either IL-1 or IL-6 therapy.
Olga, have you seen adults who developed MAS have it happen again?
I actually did have a patient have recurrent MAS, and I feel like patients who already had one episode are more likely to develop one. The question is why it happens. Could be more severe disease, more active immune system, higher levels of inflammatory cytokines, but it does happen, yeah, quite a bit in patients who have severe disease.
And I've seen it as well. So the right side of this particular slide goes into infections and neutropenia. I'd like to ask, basically saying none of those events were seen in placebo, and there were few events seen in the patients who were on tocilizumab. I want to ask Olga, Dan, and Hermine what their experience is with infections and neutropenia with tocilizumab.
Neutropenia is well known to be a side effect of tocilizumab, especially when high dosed. And when we give infusion in clinical care to SJIA children, we often evaluate the CBCs first before we decide to infuse. We do know if one decreases the dose, which is very well tolerated in many SJIA patients, then the neutropenia goes away.
Right.
We have done a study where we looked at neutropenia and the relationship to infections, and we couldn't find anything in the clinical trial data.
Interesting to note. Okay, I think we can wrap up. I wanted to ask our panelists some important questions I think the audience is probably considering, and that is, someone diagnosed with systemic JA in a kid or an adult, what's your go-to drug, IL-6 or IL-1, and how do you choose? Let's start with Dr. Brunner.
Now I have to ask you to please repeat your sentence because I was distracted for a second.
That's okay. Someone newly diagnosed and not doing well despite steroids, which are you going to choose, an IL-1 inhibitor, an IL-6 inhibitor? Do you have a rationale for one versus the other?
It comes down to a patient preference. Some families truly prefer having an IV infusible medications, and they're just like that. And others prefer an injection. And there is no rhyme or reason, but that's what it is. I put both versions out. On average, patients respond equally well to IL-1 and IL-6. However, there are about 15 to 20% of SJIA patients who only respond to IL-1 or only respond to IL-6. Unfortunately, there is no lab test one can do a priori to decide which one would be the better medication.
Right. Olga, how do you make this decision?
I think it's very specific to each patient and also at which stage in their disease they present to me. Most of the patients will present in early stages with a lot of systemic manifestation. And because of that, and also the more, like I would say, milder safety profile for IL-1, I would go for IL-1 first. But I've done quite a bit of switching from IL-1 to IL-6, either as a result of a secondary failure of the treatment or the disease progression and need for better control of articular disease. So that happens probably more down the line when it comes to treatment choice.
Dan, if you're dealing with one of these patients and there's a lot of arthritis on the table, does that change your thinking about which drug you're going to choose or what combination you're going to use?
Well, if it's a lot of arthritis in the absence of systemic features, then I'll use one of these plus methotrexate or leflunomide. Okay. So I think methotrexate still has a role, or leflunomide still have roles to play in those patients who have significant arthritis. And for many systemic JA patients over time, their systemic features kind of decrease in severity or disappear. And then what they're left with is varying degrees of arthritis. And those patients do get some benefit from TNF agents and perhaps methotrexate. When we did the TNF trials, we included systemic JA patients who had not had systemic features for a year and just had a lot of arthritis, and those patients demonstrated a response to TNF. So I think in patients in whom the systemic features have really kind of burned out and they're left with chronic arthritis, then TNF or methotrexate can also play a role in their treatment. So for new onset patients, it's strictly, very definitely focused on IL-6 or IL-1. And as Hermine said, it's based on patients' preference about frequency of injections or IV versus injections. And some families get influenced by the safety data being very clean for IL-1 inhibition.
There is a bit of a concern, I guess, for those of us that see a lot of these patients about when you should be using high dose versus usual dose. So in an adult with anakinra, it's 100 milligrams a day versus 200 a day. I always like to say with systemic JA and adult stills, they should be dosed at night because anakinra's got a 6-hour half-life, and your IL-1 peak is late at night. So why not use it then? And that's when I'll use my 100 or 200 milligrams. But 200 is either for very large or very sick patients not responding to 100. And then we have the results of the TENDER trial where it was 8 versus 12, but that wasn't necessarily based on how severe you were. That was based on weight. When do you decide to use high dose versus usual dose if we're talking about canakinumab or tocilizumab?
Well, for us, there is no higher usual dose. We have the FDA approved dose for systemic JIA, and that's the one that gets used.
So if someone's more than 30 kilograms, you won't use 12 milligrams per kilogram?
Not to start with.
Okay.
You know, the response, as you saw in the data, for kids who were more than 30 kilograms, I got treated with 8 milligrams per kilo. The response was very good. Now, what happens is after you've used it for a few months and you've got incomplete response, then there is some who will increase the dose for tocilizumab to see if we can get more complete response with higher dosing. But I think the initial dose is pretty much along the FDA approved dosage. For anakinra, there are people in pediatric rheumatology who, you know, will dose anakinra at 8 and 10 milligrams per kilo, even up to 20 milligrams per kilo. So it's been shown with anakinra there's a very broad safety profile. Most people don't start that high, but certainly we will escalate the dose above what would be the equivalent of 200 milligrams for an adult. We'll get a higher dose in kids based on milligram per kilogram.
Hermine, what do you think about dosing?
If you look, there's one pharmacokinetic study on anakinra in very small infants, and it has been shown that a dosing of 6 milligrams per kilogram in a kid under 2 is the same as a 2 to 3 milligrams in an older kid. So when we dose 12 milligrams per kilogram in small SJIA children, it is to have the same medication exposure as one would expect to block a similar amount of IL-6 in an adult patient. So you have a different pharmacodynamics in a child compared to an adult, and the doses make up for that.
Excellent. That's a really important point. Olga, do you have any approach to high doses versus usual dose when you're treating stills?
Yes. In my adult patients, I tend to use higher dose when they're in active disease state and usually taper down to lower maintenance dose when they achieve low disease activity or remission. I feel that I had more success with achieving good treatment response with higher doses initially.
Okay. I want to bring up a point that Dr. Brunner brought up earlier, which is there a role for JAK inhibition in patients with systemic JIA? I have used JAK inhibitors in patients who both during an MAS episode and as a follow-up, meaning that they treated their MAS in the hospital with high doses of expensive drugs that were not going to be available as an outpatient. And I was able to treat the patients as outpatient with JAK inhibitors. So where do you think this is going as far as JAK inhibition and management of stills or MAS?
Well, there's an ongoing clinical trial of JAK inhibitors, of 2 JAK inhibitors in systemic JIA. The results are still pending. As I alluded to, MAS is not like, it comes in different severity forms, and the earlier you diagnose it, the easier is it to treat. In those patients who have low-level chronic MAS, JAK inhibitors probably going to have a role to maintain like a very low MAS activity, so to prevent longer-term damage. So that's what we do with our very sick SJIA autoinflammatory kids in Cincinnati.
Yeah. I want to get to our questions from our audience. Dr. Fung asked a question I think we already addressed, which is when to use an IL-1 versus an IL-6 versus a TNF, and TNF when there's a TNF inhibitors when there's a dominance of arthritis. So I hope that answers your question, Eugene. Robert Quinnett in New Orleans asked a question about starting doses of in stills of anakinra, and I think we would use all of us, I think, and please group argue with me or correct me, that we use the recommended starting dose. So adults, anakinra would be 100 milligrams, and I give it at night, but if they're not responded, I'll go to 200. And that's of course in patients without MAS. With MAS, I'm going with the highest possible dose I can get away with in the hospital for either canakinumab or anakinra or tocilizumab to get rapid control. And I'm waiting for the opportunity to use emapalumab. Anybody have a different approach to starting doses?
Well, for pediatric patients with stills or SJIA, the usual starting dose is 1 to 2 milligrams per kilo. Some go higher, but that's a usual one. And if in fact the patient has MAS as part of their presentation, it's critical to jump on that with corticosteroids. I'm not comfortable with being dependent on either IL-1 or IL-6 therapy alone to treat MAS in a systemic JA patient. So in my approach, it's steroids and cyclosporin to get the disease, the MAS under control, and then perhaps you can maintain disease control with IL-6 or IL-1 inhibition.
So Dan, the other part of Dr. Quinnett's question is, do you use etoposide, and in your case, cyclosporin? I think it has to do with what other specialties are involved in the consults. If heme gets involved, it's etoposide. If a non-heme rheumatologist or someone else is going to be cyclosporin. But do you add either etoposide and or cyclosporin right from the start when you're using high-dose steroids with or without a cytokine inhibitor?
It depends on the severity of the MAS, but if the kids have neurologic or cardiovascular instability, then for sure you add it. If it's just they feel poorly and their labs are concerning, but they're still alert and cardiovascularly stable, then perhaps you can just get by with high-dose steroids. But if it's beyond that, then I think, in my opinion, it's important to add cyclosporin to the high-dose steroids.
Right.
We do use etoposide, but we try to avoid it because of secondary malignancies that are reported with etoposide. So we try cyclosporin first, but we normally layer on cyclosporin if pulse steroids have not worked. May give it a day or two and see how the patient is trending. Does the ferritin come down? Does the patient look clinically better? If that is the case, we would stay the course. But if the course is protracted, then cyclosporin would be an option. Or if things really not go well, then we would add on. We would try a JAK inhibitor. We may try other biologics, but Gamifant would be one of our next go-to if we can get our fingers on it.
Yeah, I'd find Gamifant to be way more efficacious than etoposide and relatively free of side effects too.
Yeah, Gamifant is the emapalumab monoclonal antibody. It's approved for HLH. That has had, I think, about a dozen patients with systemic JIA treated with it, and may end up with an FDA approval for that in the future. Dr. Fung asked about, he wanted a correction on what Dr. Lovell was saying. I think the answer is yes. You use IL-6 and IL-1 inhibition when systemic disease is in play. That's really what you have to, that's what you have to rely on in addition to other things. And so it's important to distinguish between systemic and articular disease, as Dr. Petryna talked about at the top of the hour. Does anybody want to comment on the use of, or is there a role for IVIG in the management of Still's disease or MAS?
Well, a very long time ago, we did a randomized controlled trial of IVIG and systemic JIA, and it did have a moderate benefit, but the benefit went away very quickly after you stopped the IVIG. So I think in the current setting with the access to IL-1 and IL-6, there's really very little role for IVIG in systemic JIA.
Which brings up a really last point that I want everyone to weigh in on. How do you know when to stop? So IVIG, when you gave it, it worked. And then when you stopped it, the things came back quickly. Still's disease by nature is a disease that will remit spontaneously and without any sort of forewarning or without any clear biomarker. When I diagnose patients and they ask, how long am I going to have to take this stuff for? I say 8 months to 8 years, and we'll figure it out along the way. But what are your rules? And we'll go around the horn here for knowing when to reduce the tocilizumab or the canakinumab or the anakinra in someone who's doing well on the therapy. Let's begin with Olga.
Yes, so typically I use both clinical and serological parameters to make that decision. I want to make sure that patient does not report any active symptoms on treatment for at least 6 months before we consider taper. And during the 6 month period, I would expect them not to have elevation of inflammatory markers, ferritin levels. In some cases, if I have my doubts, I check IL-6 levels when it's feasible. And I just want to assure that they're not having any active, an elevation of inflammatory markers, in addition to not having symptoms before we try a taper.
Okay. Dr. Brunner?
In the clinical trial, we tapered immunosuppressive therapy in a patient who remained on a given therapy inactive for at least 6 months. So I would taper the biologics after I discontinued any background medicine, like NSAIDs, steroids, methotrexate if a patient was on. Then I would not stop abruptly, but rather taper IL-6 and IL-1. That can be either done by prolonging the interval between the canakinumab injections or decreasing the dose, and the same can be done for tocilizumab. You will see that a subset of patients tolerate the tapering and a subset of those will not. And here then you have the answer. If you can't even taper it, then don't try to discontinue them. But in those who can taper it, you will continue to taper until you get to the lowest possible dose.
Okay. Dan, I think we have to cut it off there because the top of the hour, I don't want to run over. I want to thank Drs. Lovell and Brunner and Petryna for a wonderful discussion and a really good journal club. I want to remind the audience, next week or the week after, we're going to have a journal club or a Tuesday night rheumatology dedicated to discussion of new GRAPPA guidelines. So look for that in your inbox. Thanks, everyone. Have a good night.